The overall goal of this procedure is to recover infectious murine norovirus using two reverse genetic systems.
Hello, I'm Armando Arias from section of Virology in Imperial College of London. In our group, we're interested in dissecting the molecular mechanisms underlying norovirus replication. Noroviruses are a major cause of gastroenteritis worldwide, yet molecular techniques for their characterization are still relatively new. The identification of new antiviral approaches and insights into the norovirus life cycle have been limited due to their inability to complete a productive infection in cell culture. The recent isolation of a norovirus capable of infecting mice and propagating in cell culture, namely murine norovirus, has opened new avenues for the investigation of these important pathogens. In this report, we will demonstrate two strategies that allow the generation of genetically defined murine norovirus isolates in cell culture. Both strategies are based on the generation of viral transcripts capped at the 5' end. The first involves the synthesis and capping of viral RNA in vitro prior to transfection into cells. The second approach entails the transcription of murine norovirus cDNA inside of cells expressing T7 RNA polymerase.
Procedure overview. The protocol for the recovery of murine norovirus from capped RNA transcripts is initiated by linearizing the plasmid containing MMV cDNA. For MMV1, this is done with the restriction enzyme NHE1. The DNA is next transcribed in vitro into RNA using T7 RNA polymerase. Subsequently, the MMV